I hope that you found that draw along to be really helpful for Meiosis 1 and Meiosis 2. It's really helpful to draw your own set out. And what I'd especially encourage you to do is to go through and draw out prophase 2 through telophase 2 for the second set of chromosomes that I didn't draw out completely in the draw along. And along with that, I have a couple of suggestions for working through and thinking about meiosis. Check your understanding and check your memory of when did crossing over happen, or synapsis as it's called. Why not give it a fancy name? Also, make note of when the homologous chromosomes separate. Also make note of when sister chromatids separate, and making mental note that those sister chromatids are no longer identical by the time they separate. Also, check and see how many individual pieces of DNA are present in each cell at the end of meiosis 1. And then lastly, here's another question to think about. So if we ignore crossing over, in the cell we've worked with, how many different kinds of gamete cells can be created at the end of meiosis? How many different kinds are possible? All right, and another useful follow-up study exercise would be to, from memory, go back and draw out mitosis for the same starting cell. So that was that diploid cell 2n equals 4, to contrast mitosis with meiosis. Okay, so I want to come back to that second observation I started out with for this series, that parents look different from their offspring. And by now, we've actually seen 3 mechanisms for creating variation. So the first of those is the independent assortment of chromosomes. So during metaphase 1 and anaphase 1, that independent assortment of chromosomes, right? So that's the idea that the big blue chromosome and the little blue chromosome could both go in one direction, and then the big red chromosome and the little red chromosome could go in the other direction. Or it's equally likely that you could have the little red chromosome going with the big blue chromosome, and then the little blue chromosome going with the big red chromosome, right? So that's the idea of independent assortment. So the chromosomes assort independently from each other. So the second source of variation is that process of crossing over. So, swapping a piece of the chromosome with its homologous chromosome, right? So now, these sister chromatids aren't identical to each other anymore. So if there's something that's different on the red chromosome compared to the blue chromosome, now it's in association with other things that are on this blue chromosome. And that's a point we're going to come back to later. And then the last source of variation is random fertilization. So we'll talk more about this a little bit down the road, but when this cell goes all the way to the point where gametes are formed, so let's look at those gametes. So we're going to have 2 gametes that have big red chromosomes, and one of those has a blue tip, right? And then we have a little blue chromosome in those gametes. And then we have other gametes that have a little red chromosome and a big blue chromosome with a red tip on one. Okay. So you know, when a mating event happens, which of these winds up being a gamete and which one winds up going on to fertilize. So maybe this is going to be the lucky winner this time, right? Okay. Maybe it's this one instead. Maybe it's this one. Maybe it's that one, right? So there's a 1 in 4 chance that any of these cells is going to wind up being the lucky cell that gets fertilized or that does the fertilizing, but an equal chance. So the process of fertilization is random. All right, and that's it for meiosis for the moment. We're going to keep working with some of these ideas in our upcoming lectures.